Hi everyone, this is an introduction to the Yeast Lab. Background: in this lab you will use yeast and sugar to help understand a new process called cell respiration. This lab and the cell respiration document in your drive will help you answer the conclusion questions at the bottom of this lab. So you're going to do two assignments sort of simultaneously. This lab and the cell respiration assignment. Both of them are in your Google Drive. We will be assessing how well you can do the following: construct a purpose and a scientific hypothesis, identify the control group and experimental group, identify independent, dependent, and control variables, and analyze your data. Once you have an understanding of what this lab is about, you're going to write a purpose. In addition, you'll write a hypothesis as to what you think is going to happen and why. Materials: test tube rack, 3 test tubes labeled A, B, and C, graduated cylinder, beaker with water, yeast, water, sugar, a scoopula, 3 balloons, a scale, paper, string, and a ruler. Make sure you have all of these supplies. If you're missing anything, please ask your teacher. Here's what you're going to do. You're going to add 15 milliliters of water to test tube A, 15 milliliters of water to B, 15 milliliters of water to C. Turn on the scale and make sure it's in grams. Here's how to use the scale. If you forgot, click on the link and there's a short video that reminds you of how to use the scale. Place a small paper square on the scale and press the power button. Be sure the scale reads 0.0 grams, G. That G is going to be in the lower right hand corner of the scale. Make sure it's in grams. Weigh 0.5 grams of sugar and add it to test tube A. Swirl the mixture gently. Weigh 1.0 grams of sugar and add it to test tube B. Swirl the mixture gently. Weigh 1.0 grams of yeast and add it to A. 1 gram of yeast added to B. 1 gram of yeast added to C. Swirl gently after each one. Place 1 balloon over each test tube. Put the test tube in the test tube rack. Observe what happens for at least 15 to 20 minutes. Record your data. In the 15 to 20 minutes that you're waiting for your experiment to take place, you're going to do the following things. If you haven't already created a hypothesis, you should do that now. You should also know what the purpose of the lab is, but if you don't, it's okay. You'll know soon enough. You're going to click on the diagram. You're going to move the words and balloons to show the setup. Use the diagram to complete the table. So here's the diagram. Again, to edit a diagram, you click on the diagram, click on Edit. Leave the test tubes where they are and move the balloons. Move the words, okay, and tell us what is in which test tube. When you're done, hit Save and Close. When you have the diagram finished, fill out the chart. Tell us the water in test tube A, B, and C, the amount of sugar in A, B, and C, and the amount of yeast in A, B, and C. If you're confused, go back to the procedure. The next thing you'll do in the 20 minutes that you're waiting for your experiment to happen is to tell us what the control group is and what the experimental groups are. If you forgot, the same link from before is still linked. So click on the link to review. You're also going to tell us what the variables are: independent variable, the dependent variable, and 3 control variables. 3 control variables. If you forgot what variables are, click on the link. It'll open up to a new document and review independent, dependent, and control variables. At this point, your balloons should have done something, and you're going to measure them. So take the string, measure the circumference of the balloon at the biggest, fattest part of the balloon. You're going to measure it in centimeters. You're going to put your measurement for test tube A here, for test tube B here, and for test tube C here. Optional is a photo of your experiment. We'd love it if you want to upload a photo. You would take the photo using Photo Booth or your own camera, get it to the desktop of this computer, insert an image, and upload it. Or if you're using Photo Booth, you can do it right from here and take a picture of your experiment. It's optional. Describe what you see in at least 2 sentences. We don't need a whole huge lot, but we just want you to tell us, what do you see? What's the difference between test tube A, test tube B, and test tube C? And finally, the conclusion. Some of this you will be able to do right now. Other questions you will not be able to do right now unless you've already done the cell respiration assignment. So describe the experimental setup, restate your hypothesis, Tell us whether it was supported or refuted. Use your data to explain how it was supported or refuted. Yeast is a living organism that needs to make ATP. What structure does it have in all of its cells? What process did the yeast go through? From here to here, you will need to do the cell respiration assignment in order to answer these questions. Please do not Google these questions. Please do the cell respiration assignment. Thank you. Okay, that's it. Let us know if you have any questions.